Hello everyone, in this video we are going to see how to calculate specific death rate constant from decimal reduction time. Okay, first let's see what is decimal reduction time or D value. Decimal reduction time is used to measure the rate of microbial inactivation. Decimal reduction time is also known as D value. Okay, so D value is the time required at a specific temperature and under specified conditions to reduce the microbial population by 1 decimal. Okay, so 1 decimal means if your initial population is 100 means, then after your decimal reduction time you have only 10 cells per ml. Okay, so you are reducing the microbial population by 1 decimal. Okay, or in other words you can define as D value is the amount of time that it takes at a certain temperature to kill 90% of the organism being studied. Okay, after an organism is reduced by 1 D, only 10% of the original organism remain. The problem is decimal reduction time of a bacterial strain is 20 minutes. Then what is specific death rate constant in minute power minus 1 is? Okay, so specific death rate constant you can be calculated by using this formula, that is ln of xt divided by x naught is equal to minus kd into t. X naught is initial population and xt is number of population at time t, and kd is specific death rate and t is time. So here negative sign indicates there will be a decrease in the number of microbial population. So in the problem they have given decimal reduction time t is equal to 20 minutes, and here if your initial cell concentration is 100 cells per ml, then after your decimal reduction time, that is here t is equal to 20 minutes, you will have only 10 cells per ml. Why? Because here you are reducing viable bacteria by 90% at a defined temperature. Okay, so 100 minus 90 will give 10 cells per ml. So ln of xt divided by x naught, so xt is 10 divided by x naught is 100 equal to minus kd into time is your decimal reduction time is 20 minutes. Okay, so kd is equal to ln of 10 divided by 100, 1 by 10 is equal to 0.1. So minus ln of 0.1 divided by 20. So ln of 0.1 is minus 2.3023 divided by 20. So this minus will cancel it out. So 2.3026 divided by 20 is equal to 0.1151. kd is equal to 0.1151 minute power minus 1. This equation you can derive from the mathematical expression for death phase. So mathematical expression for death phase is nothing but dx divided by dt is equal to minus kd into x. So here kd is specific death rate in your death phase. Okay, so if you rearrange this equation, you will get dx divided by x is equal to minus kd into dt. If you integrate this expression, you will get integration of x naught to xt into dx by dx is equal to minus kd. It's a constant, so you can take it out into integration of 0 to t dt. So if you integrate 1 by x into dx, you will get ln x. 1 by x into dx is equal to ln x. X naught to xt is equal to minus kd into t 0 to t. So you will get ln of xt minus ln of x naught is equal to minus kd into t minus 0. You can write this equation as ln of xt divided by x naught is equal to minus kd into t. So here x naught is initial cell concentration and xt is cell concentration at time t, and kd is specific death rate constant and t is time. Thank you for watching my videos.